developing models to identify pathophysiological mechanisms and tailor therapeutic regimes according to patient-specific characteristics. So getting right down in there and modeling the patients. And so I'll let you take it away, Morgan.
Great. Well, thanks very much for the intro and for the invitation to talk today. So James kind of alluded to it. The work in my lab is really to understand individual level responses. So this is picking up a bit on both of what Jane and Allison have talked about in their talks. So we develop models of different diseases that really use a lot or look at a lot of immune responses. So we talk about cancer, or we look at cancer, but we also develop a lot of in-host models of viral diseases. And the goal in my lab is really, or one of the major approaches in my lab, is to use in silico clinical trials or virtual clinical trials. And the idea there is that we have a disease that interests us, and today I'll talk to you actually about a couple things. And then we can develop a model that, as Jane alluded to, will be complex. I'm on the side of the more is more, I guess, in terms of our modeling. But it's all calibrated to available experimental and clinical data. And then we can generate a cohort of virtual patients. And what's interesting in this approach here is that you can see these two different cohorts of patients, but they're really the same individual. They're the same parameter set. But what we can do is now look at different contexts and compare the response in the purple versus the blue to be able to identify the mechanisms that are really driving a response. And so that's what I'm going to talk to you about today in the context of our COVID-19 modeling, and also how this can be used to understand in terms of public health, why individuals might be susceptible to severe COVID-19, how this changes in the context of variants, and how this might change in the context of different patient cohorts, for example, immunocompromised cancer patients, patients as they age, patients or people who have had a prior immune response and their protection at an individual level with respect to infection. So I said I was going to talk about different diseases. I'll just go back to work that we were doing before at the start of the pandemic that continues in my lab. But it's a model of cancer treatment using viruses. So the idea here is to give a person with a solid tumor a oncolytic virus that can preferentially infect a cancer cell, and then it undergoes the same life cycle as any as a respiratory virus or other viruses, including HIV, which we also work on. And so we had been working on this story at the start of the pandemic. The idea behind oncolytic viruses, just to give you a bit of a flavor for this, is that they both infect and lyse the cells, so they destroy the cells they infect, and they also elicit this strong immune response through the normal mechanisms of cytokine secretion, a mounting of a CD4 to CD8 cytolytic response that then can also help to increase the cell destruction in cancers. So we were working on this story with my co-bourgeois at the CHUM here in Montreal, looking at co-infections of two different viruses, an oncolytic vaccinia virus and oncolytic VSV virus, and trying to understand some of the things that Jane talked about, which was when can you give both of these in an optimal way, and what are the patient-specific characteristics that differentiate responses. And so this was work that was ongoing with my former postdoc, Adrian Jenner, who's now at the Queensland University of Technology. And what it was is a normal in-host model that Jane talked about, where you have susceptible cells that are susceptible to immune pressure, cancer cells that are resistant to immune pressure, and infection by two different viral strains, the vaccinia virus, oncolytic virus, and the VSV oncolytic virus. And here we applied our in silico clinical trial approach, which is to, we had experimental data from Marie-Claude, and so we could establish ranges around the parameter values in our model, and then sample from those ranges to get out a virtual individual. So really just a string of individual parameters. And of course, you need to check that this person is realistic. And so for this, in this context, we look at the number of tumor doublings and we say, does it respond or correspond to the number of tumor doublings we would see in this specific cancer type? And if so, we accept that patient. And I won't go into lots of detail, but just to say that using this approach, and if you're interested, you can find the paper there, but using this approach, we were able to distinguish that the tumor specific growth rate, so the intrinsic growth rate of the tumor, really determined how this viral sequence, so these 2 doses of oncolytic viruses, would have an effect. And so this really showed us and demonstrated that we could segregate the population based on patient-specific characteristics that could be measured in the lab to be able to distinguish optimal treatment strategies and also to segregate out responders and non-responders. And so this is what we were working on. I'm sorry, I should have showed you that. So this is the segregation of the cohort. And so we were working on this at the beginning of the pandemic, and we had done some work previously, or I had done previous work in my postdoc on HIV. And so we felt obviously that we should flip to think about the immune response, because we're really interested in immunity in my lab as well, to SARS-CoV-2. So we haven't gone into, or Jane went into some detail, and Alison as well, about how the immune response is mounted to infection by SARS-CoV-2. So this virus will infect lung epithelial cells, and from there, these infected cells secrete interferon. And through the infection process, you have the secretion of other cytokines, including inflammatory cytokines like IL-6, GM-CSF, and G-CSF. You mount an innate immune response that can include neutrophils if things get really haywire, but really rests upon monocytes and particularly macrophages that are phagocytes. And then you have antigen that's presented in your lymph nodes that then prime CD8 positive T cells to come and destroy infected cells. In people who are asymptomatic or have mild responses, this all works quite well. There's no problem. But in dysfunctional immune responses, you have a large infiltration of monocytes, macrophages, neutrophils. You have this so-called systemic cytokine storm, where you have really high elevated concentrations of inflammatory cytokines, and you have widespread damage in the lungs because your innate immune system is going kind of crazy and the adaptive immune system can't really overtake it. And in previous beta coronaviruses, on the right here, it had been shown in some mice studies and other animal studies that the type 1 interferon response, that cytokine secreted by infected cells, is really, really important. And this was from work in the original SARS virus and in MERS. So if you mount an effective, early, and robust type 1 interferon response, you had lower viral, or animals had lower viral load. They had a better response of their T cells, and they had less inflammatory cytokines, versus those who had severe disease, who had depressed type 1 interferon responses, later responses, and therefore a higher degree of disease severity. And this very early emerged in data from China, where we saw that severe patients had increased IL-6 concentrations and decreased cytotoxic, CD4 positive and CD8 positive T cells. And so to build our mathematical model, you'll see that there are a lot of components, but starting really simply, the first thing we wanted to do was to look at, could we devise a model that could predict simply the viral loads. Originally, we had viral loads from macaques in the Oxford vaccine study. And you can see here that you have the normal kind of virus infects susceptible lung epithelial cells. They become infected and then they die and become damaged cells, which we keep track of for reasons that will become clear in a second. And you can see a delay here. This delay is related to the time it takes for cells to produce infectious virus after infection, so the eclipse phase. So that's great. We could do it for animals. We could do it for humans. So here are individual predictions of this model to individuals who were, I can't remember if they're specifically hospitalized, but they were, I think they were in Singapore and Germany. And so we had this model that could predict viral loads. Now, as I said earlier, after infection, cells secrete type 1 interferon. And so we added, sorry, where's my mouse? We added type 1 interferon into our model that effectively produces a resistant cell pool, cells that cannot become infected. Type 1 interferon is kind of like locally protective. And so those cells cannot become infected by the virus. And most of the effects in our model, we use these kinds of Hill function or sigmoidal effects models where you have some stimulatory or inhibitory on the bottom effect to some maximum effect. And this is because the model that we're using really looks at the association of molecules to receptors on cells, and this binding can be represented by sigmoidal effects. Okay, so as time went on, we had increasing information about, for example, IL-6 concentrations in mild and severe diseases in people who were interferon positive after infection or interferon negative. We had more information about T cell counts in mild and severe disease and GCSF concentrations. And so we wanted to leverage all of this into one comprehensive mathematical model. And that's what we did. So this is a lot of work. This is an ongoing collaboration that started 2 years ago now with a group of researchers. It started with Adrienne when she was in my lab. It's now taken up by Xiaoyan Deng, who's a PhD student. We had undergraduates like Sophia working with us, and we work with collaborators, including Penny and Courtney and Amber and Rosie. And what we did is we made this kind of broad model that includes the pieces I've showed you already, the viral infection, the production of interferon, and also includes the actions of the major immune players that I mentioned earlier. So neutrophils that can come and clean up virus, but that also, if there are too many of them, will cause damage to the lung cells. Tissue resident macrophages that convert into an inflammatory macrophage subset, and these inflammatory macrophages clean up debris or dead and damaged cells. Monocytes that can convert into the inflammatory macrophage subset, and CD8 positive T cells that come to really kill infected cells. And all of these actions are modulated by 4 principal cytokines in our model, so interferon, GCSF, IL-6, and GM-CSF. And we take into account the explicit binding and unbinding of these cell molecules on the cell surface. So this is a large model. Jane alluded to it earlier. Large models are complicated, they're complex, they're probably not identifiable. They do all of the bad things that you learn not to do in mathematical modeling, but we like it. We did calibrate the entire model to available experimental and clinical data. So every reaction and kinetic rate is calibrated to some information, some of it our own from Amber Smith's lab related to influenza, some from animals and other in vitro work. And when we tested our model, sorry, I can't go forward. When we tested our model, we first made sure that we could reproduce mild and severe disease. And the way we can do that is simply, and this is an emergent property, this is not something we explicitly built in, by decreasing the rate of interferon production. Decreasing interferon production, as we would potentially expect from the results from earlier SARS and MERS, you have a reduction in healthy lung tissue. You have increases in inflammatory cells, subsets, and you have a reduction in cytotoxic T lymphocytes. And of course, you have this increase in cytokines and a delay in interferon, and this increase in cytokines is really what is characteristic of severe COVID-19. So we were confident that our model could work to predict the individual response after SARS-CoV-2 infection. And so we went back to our original, or sorry, the virtual clinical trial that I kind of explained earlier, where we performed a local sensitivity analysis and identified the parameters that were most related to model outputs. That gave us a set of 7 parameters. We sample these 7 parameters within clinically or experimentally measured ranges and get an initial parameter set. We evaluate our model and perform simulated annealing to ensure that our model predictions correspond to experimentally and clinically measured outputs. So here, for example, is some longitudinal sampling of immune cells and cytokines in hospitalized patients. If our model corresponds, or hospitalized and also mild infections, if our model corresponds with it somewhere within these ranges simultaneously, we accept that patient into our virtual cohort. And so doing this allowed us to have an individual prediction of how somebody's infection might go. And we also needed to establish a way to quantify severity. And we did that by introducing an inflammatory marker, which is a measure of how severe the disease is, that looks at a number of things, including IL-6 concentrations and lung tissue damage. And here I'm just showing a paper from our collaborators where they are immunologists and they did something similar. They quantified disease severity using a cytoscore. And it turns out that a lot of their main predictors are the same ones that we see either in the disease cohort or in our inflammatory marker. And so this was kind of a nice independent verification of the work we were doing. So what do we see? If we plot in green, increasing severity, so from left to right, in each of these plots is low disease or low severity and high severity. And we plot things like IL-6 concentrations, T cells, and interferon peak shift, which I'll explain in a second. Each individual circle here, so each a vertical line corresponds to a patient. In green is their inflammatory marker. In blue is either their T cell, IL-6, or time to interferon peak. And what we found was completely, again, like I said, emergent. This was not like forced on our model, that with increasing severity, we had increasing concentrations of IL-6. We have decreasing concentrations of T cells. And very interestingly, we have this kind of distinct shift in the time to interferon peak in the patients who have the most severe disease. And we really wanted to look into this because we had this kind of evidence prior to that this interferon response is quite important. And we wanted to understand what was driving this in our model in these virtual patients. And so the first thing we did was to look at, well, what's driving IL-6 and the T cell response. And we found, again, here that interferon and IL-6 were both really quite correlated to severity. And so we wondered whether we could predict T cells given one of these. And so here you can see each individual patient colored according to their maximum T cell concentration. Darker colors show fewer T cells, and so a more severe response. You can see if you draw a vertical line that across any interferon concentration, you're liable to run into all different sorts of T cell responses, but each horizontal line really cuts through a specific T cell response. So IL-6 is really driving the T cells, or the T cells and IL-6 are probably driving each other. And with respect to interferon delays, what came out is, of course, this interferon production rate, which I kind of had alluded to earlier, which drove the mild and severe differentiation in the kind of average case. But what was really interesting is that the differentiation rate of monocytes to macrophages was really tied to this interferon delay. And that's interesting because emerging evidence shows that aberrant or like poor monocyte responses and different non-classical types of subsets of monocytes and macrophages are observed in severe patients with COVID-19. And this again was something that we didn't set out to model explicitly, but that came out from our model. And so you can read the details of this. It's been published since the summer, and probably some of you have already heard this talk. But just to follow up, because we're talking about endemic diseases now, we can use this model to also probe what could happen with respect to different variants. We can do that in a predictive way by leveraging viral sequences. So we work with Sainte-Justine and her student Fatima, who use population genetics approaches to look at viral evolution within host. And we can merge those with our model and look at and question or investigate what could drive really severe responses. So for example, increases in infectivity of the variants. This was observed early on in summer 2020 with the D614G mutant or variant, sorry, and immune modulating variants. So those that can interfere, for example, not specifically with antibodies, though that's of course very important, but for example, the interferon response. And so we do this by modifying certain terms in our model related to either infection of cells by virus or the interferon response itself. And what we can see is, I'll just go a bit quicker here. What we can see is that if we look at a range of different severe variants, so here one which has just a moderate increase in infectivity and immune modulation, or one that has a lot, kind of worst case scenario, that if we now simulate in our virtual cohort, we can characterize the proportion of severe disease in the cohort given this kind of variant. So it gives us a nice way of when we see something emerging to be able to understand what could be the potential impacts in a population. And so you can see here that variants that really modulate interferon production, for example, have quite a significant impact on severe disease and kind of more important than even infectivity, because the amount that you need to modulate production is smaller than the amount you need to modulate or increase infectivity. And so the pipeline that we use for the population genetics is available on the bioRxiv and is about to be published. And I'll just end by saying that this is ongoing work. We are working, so my PhD student Sonia has taken up some work from an undergraduate in the lab over the summer, looking at networks of immune responses in hospitalized patients with collaborators at the CHUM. And so we can use kind of data scientific approaches to make networks of interaction and communication. And Xiaoyan is now has now extended this model to look at the memory, the cellular memory response after infection or vaccination, so we can look at reinfection in a virtual patient cohort. And so I'll just end there and say thanks to, of course, the funders, but also to everyone in my lab who's worked on this and all of our collaborators, including Jane. And I really look forward to the rest of our discussion this afternoon.